All right, let's just dive right into this explainer. Look, I know finals week can feel completely overwhelming, so this session is designed specifically to be your ultimate hyper-focused study buddy. We're going to filter out all the extra noise and help you absolutely crush that massive 100-question microbiology final exam by looking directly at the high-yield topics that consistently show up year after year. Here's the game plan for our review. We're going to hit foundations and cellular terminology, high-yield bacterial pathogens, viruses, fungi, and prions, then jump into diagnostic testing methods, and finally wrap up with infection control and safety.
Okay, Section 1, Foundations and Cellular Terminology. These are the essential building blocks of microbiology.
Put a giant star next to this in your notes right now. 23. That is the exact number of linear pairs of chromosomes found in every single cell of the human DNA genome. It sounds so basic, right? But honestly, in the sheer panic of a microbiology exam, it is literally so easy to second-guess yourself when you see trick options like 46 or 3 billion. Lock it in. 23 pairs. Now this illustrates one of the most clever and honestly dangerous mechanisms in the microscopic world, a pilus. It isn't just some physical appendage, it's a bridge. Bacteria actually use this hair-like structure to share genetic material with each other through a process called conjugation. So why does this matter to us? Well, because this right here is a major player in how rapidly antibiotic resistance spreads across bacterial populations. They are literally just handing each other the survival blueprints. Crazy, right?
Notice how bacteria are divided here by their breathing preferences. This is a core dichotomy you'll absolutely need to recognize for lab testing questions. Obligate aerobes, they're just like us. They demand 20 to 21% oxygen, which is exactly equal to ambient air. But if you're dealing with an anaerobic microbe, oxygen is completely toxic to them. So to actually get them to grow in a lab setting so you can identify them, you've got to use specialized reducing media to strip all that oxygen away.
Moving right along to Section 2, High-Yield Bacterial Pathogens. Think of this as the microscopic rogues gallery.
All right, time for a classic active recall question. Imagine a patient presents with a respiratory infection causing such severe swelling in the cervical lymph nodes that it creates a super distinct bull neck appearance. What pathogen are we dealing with here? If you guessed diphtheria, you nailed it. Now, spotting the clinical symptom gets you halfway there, but the final is going to test your lab knowledge too. Under a microscope, Corynebacterium diphtheria has some highly unique visual characteristics. You're going to spot V-shaped bacilli and formations that look exactly like Chinese letters. But pay attention to this. Just as importantly, remember what it does not do. It does not bubble. Memorizing those specific microscopic visual clues, that's going to secure you an easy, guaranteed point on test day.
Okay, so the crucial takeaway for this section is keeping your Clostridium species straight. These guys are heavily tested because they produce incredibly potent toxins. You need to map the specific bacteria to their classic clinical warning signs. For example, C. perfringens releases alpha and beta enterotoxins that cause gas gangrene. Nasty stuff. Then you've got C. botulinum, which should immediately trigger a mental image of dented or bulging canned foods, leading to life-threatening flaccid paralysis of the diaphragm. And finally, C. tetani is notorious for targeting the umbilical stump of newborns, causing fatal tetanus neonatatorum. You also absolutely need to know the dangerous progression of Bordetella pertussis, which we commonly know as whooping cough. It progresses in 3 distinct stages: catarrhal, paroxysmal, and convalescent. Pay super close attention to that middle stage, the paroxysmal stage. The coughing fits here are so violent that patients run a high risk of broken ribs, inguinal hernias, and even rectal prolapse. And definitely remember your patient populations here. This infection is especially lethal to vulnerable infants under 6 months old.
Okay, let's shift gears to Section 3. Viruses, fungi, and prions, the non-living and eukaryotic threats. I've consolidated the most heavily tested viral facts right here, so feel free to pause and make sure you've got these classifications locked down tight. Remember, the absolute golden rule: viruses are non-living obligate parasites. Why? Because they are completely incapable of replicating on their own. We use the Baltimore classification system to organize them into 7 distinct groups based on their genetic makeup. For instance, HIV is famously an RNA retrovirus. For influenza, make sure you recognize the H1N1 and H3N2 naming conventions. And remember, it's what triggered the massive 1918 Spanish flu pandemic. Lastly, for hepatitis A, link it directly to the fecal-oral route of transmission, which typically presents first with jaundice.
Now, prepare to be a little surprised by this next number: 60 to 70%. That is the staggering amount of humans who quietly carry cytomegalovirus, or CMV, by the time they hit age 40. Now, while it's usually totally asymptomatic in healthy adults, it is absolutely a critical virus to know because it becomes severely opportunistic in immunocompromised patients.
Time for another quick active recall check, this time looking at fungal candida infections. These are incredibly common, but the tricky part is the terminology changes based on where the fungus is attacking. So, do you know the difference between candidemia and thrush?
Here's the breakdown. Candidemia literally means candida in the blood. It's a severe systemic infection that has infiltrated the vascular system. Thrush, on the flip side, is specifically localized to the oropharynx, often showing up as white patches in the mouth and throat. Knowing these location-based terms is just a super easy way to rack up points.
Moving on to Section 4, Diagnostic Testing Methods. This is all about visualizing and identifying the invisible. Let's look at how this builds our clinical understanding. Gram staining is an absolute cornerstone of microbiology. It's step one in bacterial identification. You simply must remember that gram-positive bacteria retain the primary crystal violet dye, meaning they're going to appear blue or purple after the final step. Gram-negative bacteria, they lose that initial dye but retain the counterstain, safranin, which causes them to appear red or pink. Why does this matter? Because this structural difference in their cell walls directly dictates exactly which antibiotics will actually work.
Now, in multiple-choice exams, you are frequently going to be asked to match the specific diagnostic test to its biological target. So, commit this list to memory. The Western blot test specifically separates proteins from blood serum. The ELISA test utilizes antisera for rapid identification. The Mantos test is your classic tuberculosis test where they inject a purified protein derivative intradermally. And over in the sterile processing department, they rely on biologic monitoring, intentionally running hardy Geobacillus sterothermophilus spores right through the sterilizer just to prove it's actually killing everything.
All right, the final stretch, Section 5, Infection Control and Safety, protecting the patient and the practitioner.
Let's test a little historical microbiology knowledge. While Joseph Lister is widely recognized as the father of aseptic technique, there was another early pioneer, Dr. Ignaz Semmelweis, who noticed something pretty striking. He saw that a specific group of healthcare providers had remarkably low infection rates among their maternity patients. Do you know who he famously credited for this success?
He credited midwives. Seriously, long before we actually understood the exact germ theory, the midwives' strict adherence to meticulous hand-washing practices proved that basic standard precautions save lives. Hand hygiene has always been, and remains today, the absolute frontline defense against healthcare-associated infections.
And that brings us to the brilliant concept of a surgical conscience. This is what ensures that patient safety remains the highest priority in the operating room. It's the moral and ethical duty of absolutely anyone in a sterile environment to immediately speak up and admit a break in sterile technique, even if nobody else saw it happen. Think about it. You can have all the scientific knowledge in the world, but without a surgical conscience, the patient is constantly at risk.
Well, we have covered a ton of ground in this explainer, from cellular foundations all the way up to surgical ethics. As you review this material one last time before your final, I really want you to think about pathogens like Pseudomonas aeruginosa. These are fatal, highly adaptable bugs that are constantly mutating their strategies just to survive and thrive. So ask yourself, how can you actively adapt and evolve your own study techniques over the next few days to absolutely master these vital concepts? Keep pushing, stay focused, and you are totally going to crush this exam.